Hello, everyone. Welcome to AWM Pitt Graduate Student Seminar. And our first speaker is Kimberlyn Eversman. She is a PhD student from the University of Tennessee, and her talk is a multi-scale mixed modeling framework of Parasitoid, Wasp, and host dispersal. Kimberlyn, you can go ahead.
Hi guys, thank you for having me here today. So like I said, we're going to be talking about parasitoid wasp and their dispersal. So now, of course, my mouse doesn't want to work. Okay, there we go. So first of all, let's talk about what is a parasitoid. So parasitoid wasps are valued for their use as an agricultural biocontrol to unwanted insects. So for example, if you have some kind of issue with whiteflies out in your field, you can actually buy a vial of these parasitoid wasps and then release them out in your field, and then they will take care of whatever that pest was. And so how they actually go about doing this is they lay their eggs either on or directly inside, like you can see in this photo right here. This is a parasitoid wasp laying its eggs inside of this ladybug right here. And so they lay their eggs on or inside of these hosts, and then the eggs develop within the host and then emerge, eventually killing the host. So if you've seen the movie Alien, it's kind of like that. But there's hundreds of thousands of different species of parasitoid wasps that parasitize a wide range of insects. And so since they're so highly specialized to whatever host they need to be able to complete their reproduction cycle, it's really great because you can release them in your field. And if you're trying to target whiteflies, you don't have to worry about them affecting other insects or organisms in the field. They're only going to be targeting and killing off those whiteflies. And then right here, we have an image of after they've emerged from this caterpillar. So if you want to release these in your field, and let's say that you wanted to model where they have spread about. Well, this is really difficult because they are very, very small. Sometimes they are less than a millimeter big. If you know what fairy flies are, that's actually one of the smallest species of parasitoid wasp. So they're very small, and then their host is normally very small. So in this image, I've circled some of the parasitoid wasp on this leaf just to give you an idea of how small we're talking about here. So they're really small, and then the other issue is that their flight is primarily wind-based, so they can actually travel for kilometers beyond wherever they were actually released from. So we have this kind of local movement around where they were released, and then this movement really far away because of the wind. So this creates a really difficult multi-scale challenge when you're trying to model this. So what our goal is we're building off an existing model for parasitoid wasp dispersal from a point release. So like I said, releasing it from like some kind of vial that you can buy online. And so we wish to determine the parasitoid wasp spatial effect on the target host species and vice versa. And then so we'd like to be able to incorporate some data from this paper from a first-time biocontrol release in Australia. So there's this paper where they released this parasitoid E. hyade to tackle this silverleaf whitefly. And so this was the first time it had ever been released in Australia. So they were able to determine, well, if anything has been affected by E. hyade, then we know that that's ours because this is the first time it's been in Australia. So we actually have some data from that. And then they also collected wind data over time and a few other things. So we'll use that wind data as well. But so we're going to focus on the parasitoid wasp and the whitefly from that paper. So if we want to model this, the first thing we need to think about is the life cycle of both the whitefly and the parasitoids. So let's start out with the whitefly here. So we have adults laying eggs, and then those eggs develop into this first instar, also sometimes called crawlers, because they have little legs that allows them to move a little bit. And when I say move a little bit, it's normally like a different part of the same leaf where they were laid. So really not much movement at all. And then they transform into our second instar, third, and then this fourth instar, which is also sometimes called the red-eyed instar, because you start to see the adult eyes developing there. But during these 4 stages here, they're really limited movement, like maybe moving to a different part of the same plant, if even that. So the main movement is coming from these adults who are able to fly meters and then can sometimes fly kilometers away based on the wind, kind of like the parasitoids. But during these 4 instar stages here, these nymph stages, they're feeding on the leaves, and so they're potentially spreading different diseases and viruses. And they're also excreting honeydew, which causes mold or mildew to grow on the plants, which inhibits photosynthesis and just overall causes damage to the plant. So then we have the parasitoid, which comes in and lays its eggs right underneath the nymph stage in these first 3 stages here. And then those eggs hatch and burrow into the instar and then develop with the instar. And so they primarily just focus on these first 3 stages here, occasionally the 4th, but it's more just these first 3 stages. So we have our parasitoid, which is really only affecting these stages that aren't moving, but the adults are the ones that are actually spreading the host population around. So that's kind of a brief overview of the life cycle of the host and the parasitoid. Let's start to talk about the model a little bit now. And so I'm going to give a brief overview of the models that we're dealing with, and then I'll go into a little bit more detail on some of them. So for each day, we're going to have 2 models. We're going to have a spatially dependent host model and a spatially dependent parasitoid model. And then so these 2 models are going to be interacting with each other each day. So when we're working on solving the host model, the parasitoid model is going to affect it by parasitizing or killing off some of these hosts in the instar or nymph stages here. And then when we're solving the parasitoid model, this is going to, the host model is going to affect it by having wasp emerge from those parasitized nymphs. And it's also going to affect whether it's also going to affect the wasp dispersal. So if there's a lot of nymphs around, they might be less likely to fly because there's a lot of availability for them to reproduce through these nymphs. So that's kind of a broad overview of the 2 models we have working here. Let's talk a little bit about the specifics some more here. So this is our whitefly host model here. And so I'll briefly go through the different parts that we have here. So we have our adult whiteflies laying eggs. And then so these eggs then develop into our susceptible nymphs. And so that's going to be those first 3 instars that the parasitoids are interested in for reproducing. From here, there's one of 2 options. They might either be parasitized, and that's going to be dependent on what's happening over here in our parasitoid model. If they're parasitized, then they die off and a wasp emerges from that. The other option is that they're not parasitized and they move into that late stage or that 4th instar. If they move into the 4th instar, then they become adults. And then from here, we have them moving around. So they're diffusing, they're advecting, being spread about by the wind and whatnot. And then we also have them dying off as well. So then this is the system of equations that we get from what I just described here. And then so we use this to determine the spatial whitefly population densities at the end of each day. So that's our whitefly model. Let's talk about the parasitoid side. So this one's a little bit more complex. So for each day's dispersal, we're going to consider that independently. And so we'll determine the change in position at the end of each day, and then we'll convolute our solutions at the end of each day to figure out what our total dispersal is. So daily movement can be broken down into one of two cases, whether a wasp is going to take a windborne flight that day or whether they're only going to be moving around locally. In either one of these cases, we can model that via drift diffusion using the Fokker-Planck equation, which we have over here, where this first term is advecting based on what's happening with the wind, and then this second term is just diffusion. And then, so what we know about the Fokker-Planck equation is if we have initial conditions of a point mass, which we do because remember, we're releasing it from that little vial, so releasing it from just that one little point, then we know the analytical solution is given by the bivariate normal distribution. So then the interesting part of this is, so for both of these cases, we know the solution, we need to figure out which one of the cases we have. So we need to determine whether or not they're going to initiate that windborne flight, whether or not they're going to decide to fly. And so environmental conditions are going to change throughout the day, and that's going to not only affect their takeoff probability, but also the wind advection they experience during that time of flight. And we're going to assume that they're only taking one flight a day. And then, so the chance of taking a windborne flight is going to be based on these four things here. So we're going to have a daylight response. So they normally only like to fly during when it's light out, not so much when it's dark out. We also have a wind speed preference. If it's too windy outside, they're less likely to fly. There's the state of earlier conditions. So if conditions were unfavorable earlier in the day, they might be more likely to fly during less favorable conditions later in the day. And then we also have the availability of whitefly nymphs. So if there's a bunch of susceptible whitefly nymphs around, they're less likely to fly as well. So over here, we have some of those functions. So this F, which is this yellow dashed line right here, is our daylight probability density, so brightest during the middle of the day. And then, so this green dotted line is our wind speed. So like I said, in that one paper, they collected wind data throughout the day. So we actually have wind data that we're basing this on. And then, so we have a scaling function based on wind speed. So when we scale this daylight based on the wind speed, we get this red function right here. And then, like I said, if it wasn't as favorable earlier in the day, it's going to be maybe more favorable later in the day. So that gives us this blue function right here, which is the probability of a wasp initiating a windborne flight, given that there's no susceptible whitefly nymphs present. So then to account for this last part here, and to actually get the probability of a wasp-initiated windborne flight, we have, we have the probability of initiating a flight with no susceptible nymphs, and then multiplied times the scaling factor, which is based on the availability of whitefly nymphs. So then that gives us that allows us to figure out the probability distribution for the change in position at the end of day D. And so this first term is whether or not they take a windborne flight. So that's our H function, remember. And then this is if they don't and only move around locally. And remember, we said that the solution to the Fokker-Planck equation was the bivariate normal distribution. So that's what we see right here. So that gives us the change in position at the end of each day. So to actually figure out what the population density is at the end of each day, what we first do is we take our solution from the end of the previous day and convolute that with our change in position here. And that gives us where they moved before we consider any sort of births or deaths or anything. So we move them and then we decide, okay, who survives from one day to the next. And then we also have wasp emerging from those parasitized nymphs. And that gives us the parasitoid population density at the end of day D. So we were able to get an analytic solution for the parasitoid equations. I want to talk a little, just really briefly, about solving the whitefly host model. So this is a reminder of what our system of host equations looked like. And so this coupled system is a bit more complex than the parasitoid model. So solving it isn't going to be as easy. And so what we actually need to do is, or what we're actually going to do is approximating it using a forward time centered space finite difference method. And so that turns into this big mess right up here. So if you're not as familiar with like a numerical approximations or anything, all we're doing is breaking up our time and breaking up our space. Excuse me. And so we're breaking up these into time steps, and then we're solving it forward in time. And so what that means is this is going to only be conditionally stable. So however fine we want to break up our time and our space, we just need to make sure that it meets this criterion right here so that our solution comes out nicely. And then, so I'm going to show you guys some solution videos here. And so what we're still working on right now, this is just kind of preliminary as results. We're still working on doing kind of some literature searches, and we still need to look at the data some more to actually get some decent and reasonable parameters of what this should look like. But so here we have our initial distribution for our eggs of our host, our susceptible nymphs, the parasitized nymphs, the late stage nymphs, excuse me, the adults, and then the parasitoids over here. And then, so this is our east and west and our north and south. And then, so what we're going to be looking at is the distribution at each day. Bit lazy. There we go. And so they're released from that point right there in the center. And then, so we start to see them getting parasitized here. We also see the host starting to spread out and being pushed by the wind over. And so this ran for 18 days. That was what we had wind data on. And so, like I said, this is using actual wind data that was collected from that paper. But we're still kind of working on the parameters, and so we're kind of still playing around with things, seeing how it looks. So if we decrease the effect of the available nymphs on the flight initiation, so make it where there has to be a higher threshold of nymphs to make the parasitoid want to stay, that means there's going to be a lot more initiating flight. So we should see the parasitoids spread out significantly more. And that's what we start to see is a lot more flying about, a lot more spread out distribution of host being parasitized. And we even start to see the parasitoids flying off the screen in this case as well with the small domain. But yeah, so like I said, still kind of playing around with the parameters that we're inputting for making these visuals. So that kind of leads into the next steps and future works. So the next step after this is we want to start to compare with data from that first time biocontrol release and do some more literature search and allow both of these things to inform parameter values here. So this is actually a map from that first time biocontrol release, where this was where they released it, and then they collected on a multi-scale data, so within 10 meters and then up to 2 kilometers as well. But kind of overall goal that would be really cool to do is, like, if someone, just any individuals have some kind of basic wind speed measuring device at their house or something, or at their farm, they could collect data from that and be like, I released my parasitoids here, and then could maybe get some kind of spread of, this is where the parasitoids spread out and this is where it affected the host and that kind of stuff. So that would be kind of like long-term goal that we're working towards. But I'd like to thank my advisor, Dr. Strickland, and like I said earlier, from the University of Tennessee, we get some funding from the Simonson Foundation. And I'd also like to thank you guys for coming. And then that's my virtual business card if you guys want to look me up or anything like that. So thank you guys.
Thank you, Kimberlyn.
Yeah.